So I've put up the Moodle page to help me remember to just make a few mentions about it. One is, so here are the PowerPoints from Monday's class, along with the key concepts that we talked about. So today we're entering the cardiovascular system. Some of this will be reviewed, but a lot of it will be new material. So we're transitioning from self-phys review to new material. So here's the key concepts list for this section. Again, it's a list. It's not a, it's not a text, and so it's a checklist of things to know and to be prepared to understand, either at home or here or both. This is just a list of some anatomy to know. It's part of the key concepts list, but I pulled it out separate for later reference, so that's not really important. So PowerPoints for today are on Moodle. I've decided to post them before class, so they will be posted before class, so you can look ahead and start to think about what is coming up. And think about, okay, what did I understand from the reading? What am I fuzzy about from the reading that I'm hoping Bob will talk about in class? What am I completely lost on from the reading and that I really need to get from class? So the PowerPoint will help. So I'm going to post them before class without the answers to any clickers or discussions, so that's not embedded. And then after class, I will post in the same place. You won't see both versions. I will overwrite this and this with the PowerPoints that have the answers to the clicker questions or other things that might be embedded. Okay, so before, it's the same PowerPoint, no answers before, with answers after. Here's the problem set text. Here is a text related to the lab. So to come to lab well prepared, you might want to skim the lab protocol that we'll be using in lab tomorrow afternoon, or tomorrow evening, or Friday afternoon. Well, both. Thursday night and Friday afternoon. Let's see, I skipped some sections. So up above, the schedule we talked about. Here is the link to YouTube for the lecture recordings. Here is the link to the Zoom that we are going to be using today. So at the break, you're going to go to your preferred Zoom location, whether it's in this building or in CST or home, and we will regather on Zoom. And here's the link to Zoom. Here is a general forum for comments, questions, suggestions. It can be anonymous, so you can opt whether to identify yourself or not. That's your option. So you're probably all familiar with how these forums work. Oh, look, somebody already put a forum posting there. I wonder who that could be. Well, it was me. And so if you click on the link to that forum post, you will find a note from somebody saying, don't forget that you have a COVID test tomorrow. Third-year students in the morning. No, yeah, third-year students in the morning, fourth-year students in the afternoon. Okay, so this will be a location for general announcements. If you have a comment about the course saying, this thing is really not working for me, can we fix it? Or could you go back over the cross-bridge cycle? I missed it again. That's a place to put that comment. a request, and you can be anonymous with that request. Question, so.
This is kind of like just a thing for the forum, but I can just say it now. Is there any way the problem sets can be due on Friday at midnight instead of Thursday at midnight?
Friday at midnight instead of Thursday at midnight.
Like my reasoning is I have a lot of things due Thursday at midnight for like my other class.
Oh, really?
And
Okay, I was thinking just the opposite. I was thinking everybody makes things due on Friday, so I'll move it to a different deadline.
and also like the material for the other half of the problem set is like today, and we have to get it between today and tomorrow. And people that have labs tomorrow don't
Okay.
have the 2 hours.
I'm okay with that. I'm okay with that. So, announcement that the problem set 1 is due on Friday night. You know, most of you are gonna have to enter CST after the desk closes and put it under my door. I'm okay with that. Just make sure it's fully under the door. Make sure your name is on it. The envelope for turning it in is available at the CST desk, so if you're done with it before CST desk closes, use the envelope, but if you're not, use the door. I'm okay with that, yeah. So, Sophie, could you do me a big favor and send me a real quick email, or just to remind me, Bob, remember, you said you would change the deadline, and I'll change it on Moodle. I'm not going to print out a new copy of the syllabus, although I might change it on the version that's posted. Other questions or comments or suggestions? Okay, so what we're talking about today is a transition from self-is into the cardiovascular system. We are going to use your knowledge from self-is or from AMP1 about muscle contraction and apply that information to heart muscle contraction, which is a little different. And then talk about smooth muscle contraction, and then get into the cardiovascular system itself. So use your knowledge of skeletal muscle physiology and apply it to heart and smooth muscle physiology. That's where we're going now. So this is a slide with skeletal muscle in mind. So let's get oriented. Here's the surface membrane, T-tubule, sarcoplasmic reticulum. Sarcoplasmic reticulum is a specialized endoplasmic reticulum. It's a specialized smooth ER that is specialized for muscle, and that's where the sarco comes from. If you see the prefix sarco on something, it almost always refers to muscle. Same with myo. And I don't know the difference between sarco and myo in Latin. So, okay. Surface membrane, T-tubule, sarcoplasmic reticulum, cytoplasm, and the thin and thick filaments that are responsible for contraction of the muscle. This is all part of the cell. So you'll remember from Monday and from previous classes that the circa, this is circa, sarcoplasmic endoplasmic reticulum calcium ATPase. This is the calcium pump that pumps calcium into the SR and is one of the components that maintains a low resting calcium concentration in the cytoplasm because calcium is pumped into the SR. It's also pumped out of the cell by the PMCA, and it is pumped out of the cell by NCX, the sodium calcium exchanger. But let's focus on circa. The concentration of calcium in the endoplasmic reticulum lumen is high because of the pump, and it's low out here because of the pump. when an action potential, a muscle action potential, which is like the ones that we talked about on Monday, voltage-gated sodium channels, sodium influx, depolarization, when that propagates into the T-tubule, it is sensed by a protein, a voltage-sensing protein that is embedded in the T-tubule membrane. This is a protein. It senses the depolarization. Your textbook calls it the DHP receptor. I call it the voltage sensor. It's also called a calcium channel, and you'll see why in a minute. Just to be clear, why do they call it a DHP receptor? DHP stands for dihydropyridine. You don't have to know this. DHP stands for dihydropyridines, a class of drugs. There are drugs that interact with this protein. And so this protein is the receptor for that class of drugs. And so a pharmacologist would call this protein the DHP receptor. A physiologist, which is what you are becoming, would call it a voltage sensor because it responds to voltage. I will accept either name because your textbook uses the pharmacology nomenclature, and I tend to use both, kind of interchangeably. Just to be clear, we're talking about the same thing. This is a drug-related name, but its function is to act as a voltage sensor. It responds to the action potential that propagates into the T-tubule, undergoes a conformational change. In skeletal muscle, this protein undergoes a conformational change. That conformational change is communicated to another protein, that is embedded in the SR membrane. The SR membrane and the T-tubule membrane come very close together, sort of molecular distance apart from each other. And the voltage sensor and this protein in the SR membrane are, in skeletal muscle, are physically in contact with each other. So a conformational change of this sensor communicates to this protein, which undergoes another conformational change, and it opens a calcium channel. So I call this the calcium release channel, because it is the channel from which calcium is released from the SR. Your book calls it the ryanodine receptor because your book is using pharmacology names instead of physiology names.
So the DHP receptor, in this case, is in physical contact with this other receptor, which you call it the calcium release channel.
Right, so let's use physiology terms. The voltage sensor of the T-tubule membrane is in contact with the calcium release channel of the SR membrane. And a conformational change of the voltage sensor causes an opening of the calcium release channel from in the SR membrane. Channel opens, calcium flows down its concentration gradient, flooding, sort of, the cytoplasm with calcium. So the calcium concentration of the cytoplasm goes from something like 0.1 micromolar at rest to something like 100 micromolar when this is excited. A big change in cytoplasmic calcium concentration. It's still low, micromolar, but it's way higher than it was before, a factor of 10,000.
And that changing concentration is just in the like cytosol or where?
This is in the cytoplasm of the muscle cell. Remember, we're focusing right now, we're reviewing skeletal muscle. The reason I'm reviewing skeletal muscle is I'm going to be using these concepts with two modifications to describe cardiac muscle and smooth muscle. So I'm starting with the review and then going to change it a little bit for the muscles that we really need to know heart muscle, and smooth muscle.
And do we need to know this for the quiz?
We need to know this for the quiz. I don't know, we'll have to, why don't you look at the quiz, when we're looking at the quiz together in our Zoom meeting, because that's what we're gonna be doing during the Zoom breakout.
Okay.
Let's see if that's true.
Okay.
I don't have the quiz completely memorized. I think the answer is no, but I don't want to promise.
Okay.
Calcium, now you hopefully remember from cell phys that calcium, cytoplasmic calcium, is the trigger for the cross-bridge cycle of the muscle cell. So cytoplasmic calcium triggers the contraction of the cell. At the end of the contraction, calcium is pumped back into the lumen of the SR. Calcium falls, calcium concentration falls, and relaxation can occur. That's review. Everybody okay with that so far? The skeletal muscle and cardiac muscle are both considered striated muscle because they look stripy under a special microscope. Striped. That's what striated means. And so the sarcomere of skeletal and cardiac muscle is formed from a repeating unit of Z discs connected to thin filaments, M lines in the middle of thick filaments, and thick filaments and thin filaments interacting with each other to do the contraction. There are bunches of other proteins. For example, this big protein called titin. There's other proteins, one of which is called dystrophin, which is named for the disease that it triggers when it's mutated, called muscular dystrophy. So there are other proteins here, but these are the key players. And in fact, it's more than the key players. You don't really need to know titin. What you need to know are actin thin filaments connected to Z discs and myosin thick filaments in a opposing perspective. So the head groups of myosin on this side are reaching out this way, and the head groups of myosin on this side of the M line are reaching out this way. So the myosin thick filaments are anti-parallel. So these bundles, all of these myosins are bundled together, all of these myosins are bundled together, and they're opposite on either side of the M line. For this course, I don't need you to know all the nomenclature of the sarcomere. That's the other thing, of course. Okay, so let's do the cross-bridge cycle, which we did in cell phys, and we did in AMP1, but let's go through it. So this is from your textbook. Let's get oriented. Here is a Z line, so I'm going to go back and forth because this cartoon is based up, so this is data, this is a cartoon, and this is a cartoon, okay? So here's a Z line, or a Z disc, they're the same thing. And let's see it. Which one of these is this in a moment here? Okay, so this is analogous to, this Z disc is analogous to this one here. So this Z disc is connected to a thin filament and a thick filament and only one of the head groups of this bundle of myosin is shown. But there's hundreds of them here. It looks much more like this. This is what it really looks like. This is a good cartoon of it, and this is now a highly simplified cartoon of it. at rest, when the muscle is relaxed, can be stretched. Heart can be stretched, skeletal muscle can be stretched. When at rest, the calcium is low. Calcium is not bound to troponin, and so the troponin and tropomyosin of the thin filament prevent the myosin from interacting with the actin. Let me say that again, because that is the key. We'll get oriented. Let's get oriented. Myosin, thick filament with the head groups. Actin, thin filament with troponin and tropomyosin. Troponin is the pink blobs and tropomyosin are the blue ropes. And actin are the gray spheres. In low calcium, at rest, troponin and tropomyosin block the sites on actin that the myosin head group would interact with, if it could. But it can't, because it's blocked. So the thick filaments and the thin filaments are not attached to each other, and that's what allows the muscle to get stretched, because they're not physically attached to each other. This Z-disc could get pulled this way because this attachment is loose, and this Z-disc could get pulled this way because this attachment is loose, and so the muscle could get stretched. Okay? Now we contract it. Now we're going to start the contraction. Calcium goes up. Remember, action potential, voltage sensor, calcium release channel. Calcium goes up. Calcium in the cytoplasm goes up because it's being released from the SI. Calcium concentration rises. Calcium binds to troponin and causes a conformational change. That conformational change of the troponin and tropomyosin expose the sites on actin that myosin can bind to. So myosin binds to it. The binding of myosin to actin triggers a conformational change in the troponin, in the myosin, causing it to release. So this is, what is this? This is actin, myosin, and the myosin is bound to ADP and phosphate. You'll see where that comes from when we complete the cycle. So the actin is bound to myosin, and the myosin is bound to ADP and phosphate. When the myosin binds to actin, the affinity for ADP and phosphate goes down, way down. And so ADP and phosphate are released from myosin. That release causes the power stroke. The power stroke is caused by the release of ADP and phosphate. So this head group moves from an extended conformation to a flexed conformation. And since it is attached to the actin, it pulls the actin. And so it's not just this one myosin head. All of these myosin heads are binding and pulling. And so this Z disc is being pulled this way. And this Z disc is being pulled this way. And we now see contraction. The power stroke. But it's a cycle, so we gotta complete the cycle. After the power stroke, the nucleotide binding site is empty. because ADP has been unbound. ATP binds. ATP in a healthy muscle is high. The concentration of ATP is high. So ATP binds to the myosin. The binding of ATP to the myosin triggers a conformational change of the myosin, lowering its affinity for actin. And so myosin lets go of actin. ATP binds, so we have actin, myosin, ATP, but this is not a dot here, this is a plus sign, because the actin is not bound to the myosin. The ATP has triggered the myosin to let go of the actin. The myosin hydrolyzes ATP. The myosin is an ATPase. So the myosin hydrolyzes ATP, and now we're back to our starting point. Myosin not attached to actin, ADP and phosphate bound. And so it can, by going through repetitions of this cycle, the thick filaments pull on the thin filaments, because even though this, this looks like it's going to the same spot, this myosin is binding to the same spot, but it's not. It's binding to a spot closer to the Z disc, because the previous cycle moved it. Okay? So the cross-bridge cycle, triggered by calcium, causes the myosin to pull the actin thin filaments, shortening the sarcomere, causing contraction of the muscle. That's the skeletal muscle cross-bridge cycle, which I do think is part of your quiz. All right, problem set.
Is this when the ATP is hydrolyzed, is that when myosin goes to the extended position?
When ATP is hydrolyzed, myosin goes to the extended conformation. And if I didn't say that before, I apologize, because that is what ATP hydrolysis does. ATP hydrolysis causes the myosin, which was previously flexed, that is now let go from the actin, to re-extend so that it can bind to a new site on the thin film.
So, for the ATP binding to the myosin head,
ATP binding to the myosin head.
yeah, so does it do two things? So the first one is the actin detaching from the myosin.
That's what the binding does.
Okay, and the second one is from bend to straight.
That's what hydrolysis does.
So the hydrolysis causes it to go from bend to straight.
Goes from, let's say, flex to extend.
Sure, yeah. And for the binding of ATP, that causes it to detach from the actin and myosin.
Right.
Detach.
The binding of ATP causes it to detach from actin. The hydrolysis of ATP causes it to go from flexed to extended.
Thank you.
And multiple combinations, doing this 100, maybe 1,000 times, happens per contraction and pulls the Z discs short. Okay, so now let's get to heart muscle. New material. Heart muscle is striated like skeletal muscle. So all this image here, which you are forming for skeletal muscle, works for heart muscle, cardiac muscle. You may remember from cell phys that heart muscle cells are big cells. Like one of the muscle fibers in my biceps is this long. It's not very wide, it's not very thick, because I've got thousands of them. But each of them is this long, because it's formed by the fusion of thousands and thousands of myoblasts, the cells in development. So as my body was developing, myoblasts, precursor cells to muscle, fused and made this one gigantic cell. Heart muscle does not do that. Heart muscle cells are individual cells. They're cellular in dimension. They are not inches long. They're microns long. They're what you think of as a cell. But they are connected. So they are connected to each other. So this is supposed to be a cartoon of a cardiac myocyte, muscle cell. And it's got this funny Y shape, but they're not all like that. Most of them are shaped like little bricks. Some of them are Y-shaped. The point is these cells are physically and electrically connected to their neighbors at a specialized structure called the intercalated disc and the desmosome. So that's jargon, that's names. So the intercalated disc is the name of the physical connection of one heart muscle cell to its neighbor. And the desmosome is a specialized name for where the electrical connection is between one heart muscle cell and the next. So they're physically connected at intercalated discs, and they are electrically coupled at desmosomes, which are within the intercalated discs, because of channels that span one membrane to the other. So channels connect this cell to this cell. It's a separate cell, but it's got channels that connect the cytoplasm of this cell to the cytoplasm of this cell. And they're called gap junction channels.
Now, why would we connect this cytoplasm?
You'll see. The short answer is because when the heart contracts, we want the whole muscle to contract at the same time. We don't want this part to contract separate from this part. We want the muscle to contract in a unified, consistent, time-organized way. And so we evolved a mechanism for the heart muscle to contract more or less simultaneously. Yes?
Could a desmosome be equivalent to like a synapse? Or should I try to not?
It's equivalent to an electrical synapse,
Okay,
but it is nothing like a chemical synapse. There's no chemical connection here,
right,
but it is an electrical synapse
Okay.
between the two muscle cells.
okay.
So if you know anything about electrical synapses, just take everything you know about electrical synapses and apply it here.
Okay.
But it's not a chemical synapse.
Thank you.
Yeah.
Can you repeat what the gap junction stuff?
The gap junctions are ion channels that span two membranes. The membrane of one cell and the membrane of the next cell. And so the ion channel connects the cytoplasm of one cell to the cytoplasm of the other cell, the connected cell. So let's do a quick. So I see some fully correct answers, I see some partially correct answers. So is cytoplasmic calcium required for myosin to bind tightly to act? Yeah. Is cytoplasmic ATP required for myosin to bind tightly to act? No. Cytoplasmic ATP is required for myosin to let go of actin.
But if it's not free, can it not bind again?
So you're going to the next cycle. That's what, so after hydrolysis of ATP to ADP, that's what allows the myosin to bind tightly to actin again. So if you say, well, ATP is required to get ADP, then I sort of understand where you're coming from. But by itself, ATP is not a correct answer. It's only correct when you're thinking, okay, I need ATP to get ADP. But that's not the only way we can get ADP. So the strictest answer is A and C. If you chose all of the above, and then were able to explain to me why you chose ATP, I would probably accept the answer. If if you said, I chose ATP because that's how I get to ADP, and then if I choose A, B, and C, that's the same as D, I get it. Okay, which of the following is required for the power stroke? Good, I'm seeing a lot of red, blue. Red, B. So ADP and phosphate dissociation are required for the power stroke. Okay, good. So some review of the cross-bridge cycle. Now let's turn to smooth muscle. Why do we need to talk about smooth muscle when we're talking about the cardiovascular system? It's pretty clear why we need to talk about cardiac muscle, because that's the muscle of the heart. But we need to talk about smooth muscle because this is the muscle that controls the diameter of arteries and veins. And the diameter of arteries and veins is going to be important for determining blood pressure. Okay, so smooth muscle is different from heart and skeletal muscle. It's not striped, so it's not striated muscle. It doesn't have sarcomeres or T-tubules. It doesn't have it does not have this organized structure. It does have actin and myosin, but they aren't organized into sarcomeres and Z discs and all that. They are, in general, connected by gap junctions. So one smooth muscle cell is connected to its neighbor electrically as well as physically. They don't have desmosomes, but they are physically and electrically connected. And mostly, your smooth muscle is not under your conscious control. You can control your skeletal muscle. You can't really control your heart muscle either, and you can't control your smooth muscle. They are controlled. The rate of your heart muscle contraction is controlled by your nervous system, but it's part of the autonomic nervous system, not your conscious nervous system. So, smooth muscle lines the tubes in your body. And the ones we need to know right now are arteries and veins. And in a couple of weeks, we're going to learn about the smooth muscle that lines your airway and your respiratory system. And a few weeks later, we're going to learn about the smooth muscle that lines your digestive tract. We're not going to talk about the urinary bladder, although it happens. We don't talk about the uterus in this course, but smooth muscle, this might be the strongest muscle in your body, in some of your bodies, not in all of your bodies. Some of you have a uterus that has smooth muscle that is extremely strong. The ciliary muscle in your eye is smooth muscle, the thing that controls the pupil diameter and the tension on the lens is smooth muscle. Anyway, the ones we're focusing on right now are the smooth muscle that controls the diameter of arteries and veins. Smooth muscle, okay, so this is an actual micrograph of smooth muscle. So smooth muscle is typically organized with longitudinal fibers and circular fibers. So these are smooth muscle cells caught in cross section. So if this smooth muscle cell is coming out like this, and this smooth muscle cell is running like this, and the tube, let's just say the artery that this smooth muscle is lining, is down here. So the tube is lined by longitudinal and circular smooth muscle. In arteries and veins, the smooth muscle that's most important for controlling the diameter is the circular smooth muscle. Because you can see, if this muscle contracts, if this is a donut of smooth muscle surrounding a tube, then contracting this smooth muscle is going to narrow the diameter of the tube. And here is actually more, so this is the circular and the longitudinal smooth muscle of some other smooth muscle bed surrounding some other tube. I don't even remember if this is artery, vein, or uterus, or digestive tract. The concept is the equivalent. Smooth muscle does not have sarcomeres, does not have Z-discs, but it does have actin and myosin that do the same thing as in skeletal and cardiac muscle. That is to say, the cross-bridge cycle exists, but the thin filaments are not connected to Z-discs. They're connected to dense bodies that are anchored to the membrane. And so when a smooth muscle is relaxed, it can be stretched. And when a smooth muscle contracts, the dense bodies are pulled together and the muscle gets shorter and thicker. So the muscle contracts, but it's not as linear as it is in striated muscle. But the principles of the cross-bridge cycle are the same. Calcium, troponin, etc. Actually, no, the troponin, so I'm going to get to a difference in a few minutes. Calcium is the trigger, but the next mechanism is a little different. No?
So for the, I guess the cross-bridge cycle before you begin, do we also have acetylcholine in smooth muscles, or do we have a different kind of mineral?
So we don't have to worry, so in neither of these cases do we have a synapse, a neuron, so heart muscle contracts without a neuron telling it to contract. Smooth muscle contracts without a neuron telling it to contract.
So no neurotransmitters?
We are going to talk about neurotransmitters that modulate, but we do not have to think about a neuromuscular junction the way we think about it in a skeletal muscle. So you may know that neurotransmitters modulate the function of your heart, right? When you're stressed, the neurotransmitter epinephrine and norepinephrine cause your heart to beat stronger and faster. We'll talk about that mechanism today. No, probably on Monday. But your heart beats even if your autonomic nervous system is disconnected. Your heart has an intrinsic contractile mechanism. And same with smooth muscle. So smooth muscle contraction, so we're getting into smooth muscle contraction. It starts with cytoplasmic calcium, just like striated muscle. But I was wrong when I said troponin. The calcium in smooth muscle does not bind to troponin. It binds to another protein called calmodulin. Calmodulin is another, so troponin is a calcium binding protein of striated muscle. Calmodulin is a calcium binding protein of a lot of cells, including smooth muscle cells. Calmodulin is now activated by the binding of calcium. The activated calmodulin interacts with a kinase and activates it. So how many kinases have we talked about so far? We've talked about PKA. You learned about PKC as part of the study for the problem set. Now we're introducing a third called myosin light chain kinase, MLCK. Myosin light chain kinase is activated. So what activates PKA? Adenosine? Well, cyclic AMP. What activates PKC?
The
I3 and DAG.
DAG and
calcium.
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 Calcium and diacylglycerol are what activate protein kinase C. IP3 is what triggers the release of calcium from the ER. But it's the calcium and the diacylglycerol that activate PKC. Myosin light chain kinase is activated by calmodulin, that is activated by calcium. So what is the substrate of myosin light chain kinase? Well, it's built into the name. It's the myosin. It's a enzyme that phosphorylates myosin. And the phosphorylation of myosin in smooth muscle is what triggers the cross-bridge cycle. So the calcium does not have to interact with troponin. What happens is the kinase activates the myosin by phosphorylation. It's a different kinase. I mean, it's a kinase, and it's a different form of myosin. So the cross-bridge cycle is initiated by the phosphorylation of myosin. But the steps of the cross-bridge cycle are the same as with skeletal muscle. Binding, dissociation of ADP and phosphate causing the power stroke, ATP binding causing unbinding, ATP hydrolysis causing it to reach out again. There are thick filaments with hundreds, if not thousands, of these head groups, not just one at a time. And so the smooth muscle cell gets shorter. Calcium, if calcium is pumped by the CIRCA or PMCA or NCX, then the calcium concentration goes down and the whole thing lets go.
So you could say that both the skeletal muscles and smooth muscle cells, the contraction is triggered by calcium.
Yes, that's true.
But in smooth muscle cell, you go through this additional step to then do the cross-bridge cycle.
Well, in both cases, the calcium is doing something. In skeletal muscle and cardiac muscle, the calcium is triggering the conformational change of troponin and tropomyosin.
True.
In smooth muscle,
It doesn't call much.
there is no the binding is not to troponin. The binding is to calmodulin, which activates the cross-bridge cycle by phosphorylation.
Calmodulin, is it a protein or is it a messenger?
It is a protein. Calmodulin is a protein. And so is the kinase. So
Does the myosin have an extra phosphate group where the ADP plus the phosphate comes from the phosphorylation?
so when the myosin is phosphorylated, it has a phosphate attached. It's not shown in the cartoon.
So as
And so when the
ADP
phosphate
by
is
itself-
attached, I'm sorry?
It has ADP but not the phosphate group yet then?
Ah, okay, so in this case, it has, so in skeletal muscle, the phosphate is bound to it, but it's not a covalent binding. In smooth muscle, the phosphate is covalently attached by the kinase. And so only through dephosphorylation can the thing relax.
Oh, okay.
So both require ADP and phosphate. So, I mean, they both require ATP. The ADP is not an important player in this story, but it is an important player in striated muscle. The phosphate is important in both, but in striated muscle, the phosphate is simply bound to the myosin, and in smooth muscle, the phosphate is covalently attached by the kinase. Okay, so the next couple of slides are an overview of the cardiovascular system. Let's take a 5-minute break just to catch our breath, stretch a little bit, and then we'll pick up in 5 minutes with a finish. So what we're going to do is we're going to finish this section of the class, starting in about 5 minutes. Then we're going to take a longer break and you're going to go to your Zoom locations. We'll all be together